Congratulations, you've made it to Friday. That is, it is Friday if you're watching this on the regularly scheduled lecture day, which would be March the 8th. And hopefully at Nationals we have now sewn up a couple of victories, fingers crossed. So I don't have a lot of announcements today. I want to remind you that in fact homework 6 is due next Tuesday. But I thought since I don't have a lot of announcements to make, it would be a good time to remind you that your syllabus is really a good resource when it comes to scheduling. I think sometimes it's easy to forget that the syllabus exists out there and that in fact it does have a schedule on it. So I thought I'd pull to your attention that we're on the week of March 4th through the 8th, and you'll notice there I have announced even at the beginning of the semester that the lectures for this week would be vodcast. Scanning down there, this helps you to keep track of what's happening that week and the next weeks to come, and to remember that in fact we do have some things that are coming up. The poster presentation abstract, we're hoping we can start to get that ready to go at the end of March, and the exam 3 is tentatively scheduled for April the 2nd. So just looking ahead at the calendar of events to happen, this is a good, helpful resource for you. I did have somebody ask me in one of the groups when they sent in their initial poster topic, had asked me about the possibility of moving exam 3, and I don't know what the rationale was there. So if you would like to comment on that, or perhaps send me your thoughts on why that should be relocated, I certainly am more than willing to hear that, because you guys have done such a phenomenal job on the exams that I would be remiss not to be flexible in any way on that. That being said, let's jump into our lecture for today, which is wicked exciting because we are going to be talking about translation. So it should be quite a wild ride as we look into the process whereby RNA, mRNA specifically, is converted into protein product. Before we go there, though, I do want to make sure that we fully understand transcription, and that after having our lecture last time, we have a good feel for the process by which DNA is used to make RNA code. So in recognizing the -10 promoter region, this particular picture doesn't show all the way upstream to the -35, but we see our TATA box here, see TATA at, and we see our +1 transcription start site. It's really important to recall that transcription doesn't start until the +1 transcription start site. So this is where the actual process of making RNA from DNA begins. So that being said, let's see how good of a job we can do at making good RNA polymerase enzymes, recalling that as RNA polymerase begins to synthesize the RNA, it always pairs with the template strand. So as the newly synthesized RNA gets made, it will pair transiently with the T, T, G, C, C. So this region will get opened up. And in fact, maybe we should just go ahead and draw that out so that everybody can see what's going on here. We've got our TATA at sequence, and then we've got GCATT, and then AC, and then this is where RNA polymerase is starting to denature and unwind a region here, C, right? This is opening up that transcription bubble. And then if we draw the template strand, we've got 3', and of course that's complementary, so it will be A, T, A, T, T, A, C, G, A, or sorry, sometimes my base pairing rules are just not with the program. So A will pair with T, and then we start opening up this replication bubble, so A, and then T, G, T, T, G, right? And this replication bubble has opened up so that beginning with the +1 sequence, so right here, the template DNA is going to begin to pair with the RNA. So RNA will start to get made, and this will be A, C looking like the coding strand. So this is going to be the 5' end of the RNA. And then this is just going to continue on down the strand. So we can see that our RNA is looking like the coding strand of DNA. But we know once it gets down here to where those T's are, U's are going to replace the T's. So we can write out the full RNA. We see the beginning of it there as 5' A, C, G, G, C, and then UUAC. And I'll let you guys keep on writing to get the full sequence of that resulting mRNA. So I hope that really makes a lot of sense, and I promise I'm going to give you a lot more to practice on the homeworks, and you'll also see a question definitely like this on the exam, and many exams to come, because this is such a huge and big, you know, just a big take-home message. So as we proceed into translation, after having talked about transcription, recall that this is the process of taking the mRNA that resulted from transcription and converting it into protein product. So notice that the mRNA has a central region that we call the coding region, but to the left and right, or upstream and downstream of that coding region, we see several non-translated regions. That is, regions that are not converted into protein code. So there's an upstream and a downstream non-translated region. We call the upstream region the leader sequence, and then the downstream sequence is called the trailer region, leaving that center section to be converted into protein code, and of course that's called the coding region. Now if we give this some thought and ask ourselves, why would there be a non-translated leader sequence? Why would there be a non-translated trailer region? There's a couple of answers, and I'm picturing Jen answering this question and saying a big reason for having this trailer region is because nucleases like to chew on mRNA. And so the nucleases, thinking of them as like little Pac-Man enzymes chowing down on the 3' hydroxyl of this mRNA. If there's a non-translated leader region, the Pac-Man enzyme is going to do a lot of chewing before it ever gets to anything that matters. So it can practically stuff itself before it gets to the coding region and would actually cause any damage to that coding region. The non-translated leader sequence also has a protective role, but we can also add to that it is in this region, this non-translated leader region, that we get the area that serves as the initiation site for translation. So just as there was an initiation site for replication, the ORAC, and the promoter region for transcription, we have a non-translated leader sequence that has the initiation sequence for translation. So as we head into translation and we think about the synthesis of a protein from the mRNA, it's really informative for us to do just a little bit of review in remembering that the protein is going to be comprised of amino acid subunits. So let's reflect upon the structure of an amino acid with its central carbon, and of course its R group, its hydrogen, its amino, and its carboxyl. Recall how in each case nucleic acids were synthesized 5' to 3'. They had a directionality of synthesis. So too is true of the synthesis of proteins. They have a directionality of synthesis as well. They're always synthesized in the amino to carboxyl direction. That is, new amino acids are added onto the open carboxyl. Now, I have a pet peeve, and that is that sometimes when students are writing the sequence of amino acids in a protein, they put a 5' and 3' end on that sequence of amino acids. That is wrong. So try to float my boat for me and not put a 5' and 3' end on your proteins. Proteins have amino and carboxyl termini, not 5' and 3' ends. So let's just take a note that when a new peptide bond is formed and a new amino acid is added on to this existing amino acid, let's draw that out and recognize that now, building from the open carboxyl, a new amino acid can be added in this chain. So now we clearly have a dipeptide. But once again, another amino acid could get added onto that open carboxyl end. And once again, we get the synthesis of another peptide bond. With the addition of a third amino acid to this chain. But no matter how long the chain gets as synthesis occurs, we always have an amino terminus and a carboxyl terminus. And that is what we should always write on any of our protein sequences, is amino end and carboxyl end. So now let's talk about the process by which the RNA is read to give the corresponding polypeptide chain. So during translation, amino acids are added to the carboxyl terminus of a constantly elongating chain. We know that the mRNA is read to allow for the synthesis of that polypeptide chain. But this is where it gets kind of tricky, because remember that there are 4 different nucleotides, G, C, U, right, and A. So we know that there are 4 different bases and 20 different amino acids. 20 common standard amino acids in a protein, and yet there are 4 different nucleotides. This doesn't add up. So when we think about an RNA trying to dictate the synthesis of a protein, this just doesn't add up. So what it means to us is that the RNA must have to get read in combinations. Now we could think, okay, well, what if it gets read in duplicates? That is, 2 nucleotides get read at a time. Well, that would give us a possibility of 16 combinations, and still that doesn't add up to 20. So it has to get read in triplicates, in fact, in 3 nucleotide sequences, and many of you know that those are called codons. So an RNA is read in 3 nucleotide sequences called codons. So this works out because 4 raised to the 3, that is to say there are 3 nucleotides per codon, and there's a total of 4 nucleotides. This gives us 4 raised to the 3rd, or 64 possible codons. That works out because there are 20 amino acids. We now have plenty of codons to specify the 20 amino acids, but we actually have way more than we need. So what that must mean is that there's more than one codon that can specify an amino acid. And in fact, the sexy term for that is degeneracy. There is a great deal of degeneracy in the genetic code. More than one codon that can specify an amino acid. Of course, there are also some codons that don't specify any amino acid at all, and instead those are termination codons. So they specify stop. So let's jot down that 3 of the possible 64 codons, UAA, UAG, and UGA, encode for a stop in the production of a polypeptide chain. So these are stop codons. The other 61 codons, however, all do code for amino acids, and that's where we can see that there are many more than one codon that specifies an amino acid. This is degeneracy, and we say that the genetic code is degenerate. So that's kind of a cool way to talk about the genetic code, recognizing that there is more than one codon that can encode for any given amino acid. Thus, we have what is called a codon usage table that enables us to determine what codon specifies what amino acid. In this codon usage table, you can see already that certain codons, such as UUU, UUC, both specify the amino acid phenylalanine. Now look at the degeneracy going on here for leucine. CUU, CUC, CUA, and CUG all specify leucine. Lots of degeneracy going on there. Now, we could further note that the first two nucleotides in that codon that encodes for leucine are the same. So notice that the location of flexibility is actually in the third position of a codon. That is the most 3' side of that codon because, of course, RNA is read from 5' to 3'. So the flexibility or the loosey-goosiness in a codon comes in the most 3' position, the third one over. Let's finish filling out this table, taking note of degeneracy. The AUG codon, this specifies methionine, but it also specifies a start. So in the prokaryotic world, when an AUG codon is met for the very first time in a sequence, it actually specifies a special amino acid called N-formal methionine, but it also triggers the beginning of translation. So AUG is the start sequence. In a prokaryote, the amino acid that first gets incorporated, so that is the first amino acid in every polypeptide chain, is what we call N-formal methionine. So of course we recognize that that is the R group methionine, but then attached to the amino terminus of that methionine is a formal group. Looks like this. So that formylation on the amino terminus is always present on the first amino acid in a prokaryotic chain. So AUG specifies the beginning of translation, and then depending upon what codon is encountered next will dictate the second amino acid in the chain. So if we write down and fill in the rest of the table, we begin to see a lot of degeneracy. We see not only the stop codons, 3 of them that can all encode for stop, but also notice here, UCU, UCC, UCA, UCG all code for serine. Likewise, many codons encode for proline. We can see degeneracy in the code for histidine as well. And one kind of cool thing is to notice that in fact sometimes similar codons encode for similar types of amino acids. So recalling that in fact glutamate and aspartate are acidic amino acids, when we notice that GAU, GAC, those are aspartate, but then GAA and GAG are glutamate. So similar amino acids, similar codons that do specify them. We should also practice using the codon usage table, so we'll get good at it. So let's just start out with what amino acid is encoded by the codon AAG. So AAG. Well, first how we do this is to look at the first position, which we know is A, second position also A, and then third position G. So that allows us to come right over to lysine. Lysine, that awesomely basic amino acid, is specified by AAG. Let's practice one more. How about GGG? So G, G, G. Glycine, the tiniest amino acid, is encoded for by GGG. Awesome. So this gives us an idea of how to use the codon usage table. It also shows us in the context of that table just how degenerate the code is, just how many different combinations can come together to form each amino acid. So there can be in many cases 4 or often even more than 4 codons that specify the same amino acid. Okay, now that we've understood degeneracy, it's important to understand that the genetic code also is what dictates the start site for translation. That is, translation doesn't begin until the first AUG start codon is encountered. That is, that everything is untranslated leader sequence until the first AUG is found. We say that AUG sets the reading frame. It specifies which of the possible reading frames is the right one. So looking at this particular mRNA sequence, we need to look for the first AUG start codon before we begin reading in triplicate. So if we scan down from the 5' end looking for the first AUG, there it is there. This AUG sets the reading frame, and it's not until you encounter that first AUG that you begin reading. From there, everything can be boxed in triplet heads, and we can recognize that each one of those codons dictates another amino acid. And we could use our amino acid table to look that up. Of course, we don't need to look up AUG. We know that OS always specifies N-formyl methionine, or F-met. But we could look up this one, CGU. So C, first position, G, second position, and then U. And it looks like that's the pirate amino acid, right? Arginine. So noticing that the AUG sets the reading frame. Also, it's important when you write your protein sequence to make sure that you're specifying that this is the amino terminus, even though in this case it's formulated. And then eventually, after reading all of these codons, we'll end up with the carboxyl terminus. Wonderful. So not only does the genetic code need to be read in triplicates, it needs to be read correctly in triplicates. So always beginning with the AUG start codon. Before we begin with the initiation phase of translation, I want to make sure that we properly introduce all of the players in the process. Ribosomes are arguably the most important player because they are the factories of synthesis. They are what is synthesizing the peptide bonds. So a ribosome moves along an mRNA, reading the codons one at a time and matching each codon with the correct amino acid. So the ribosome moves along, presenting the codons and allowing for the synthesis of the peptide bonds that form between the amino acids. Now, there may be a lot of ribosomes in any given cell at any given moment all doing this job. And in fact, it's pretty cool to think about the fact that there might even be more than one ribosome reading the same mRNA at the same time. That's called a polysome. It's pretty crazy. And in an E. coli cell, man, you could have a lot of polysomes because you could have many ribosomes reading the single mRNA and many, many mRNAs getting translated at the same time. We also should remember that in fact it's an RNA within the large ribosomal subunit that actually does the synthesis of the peptide bond. That is, it's an RNA that acts like an enzyme, and you recall that's called a ribozyme. So if you're really jazzed about ribozymes and you want to do a little bit more dorking out with this, I suggest going to this URL where there's an awesome holiday lecture by a man named Thomas Cech. So let's just review the structure of the prokaryotic ribosome, 70S ribosome. Large ribosomal subunit is the 50S. Within that are the 5S and 23S rRNA, as well as 34 polypeptide chains. Now within the small ribosomal subunit, we see the 16S ribosomal RNA as well as 21 polypeptide chains. The 16S rRNA is arguably equally as important as the 23S. So that 23S rRNA is the one that's actually acting as ribozyme and making the peptide bonds between the amino acids in the elongating chain. The 16S ribosomal RNA is actually what allows for initiation of the process of translation because in fact there is a region on the 16S ribosomal RNA that is actually complementary to the initiation site on the mRNAs. tRNAs are what Francis Crick liked to call adapters, and that is such a fitting term for a tRNA because tRNAs on one end recognize the codon on the mRNA, and then on the other end they adapt that codon to the amino acid that they carry. So two very distinctive sides, sort of a polarity to this tRNA, and allowing it to be an adapter. Plug into the codon on one side and plug into the amino acid on the other side, adapting the codon to the amino acid. There are several different components of this tRNA, and do recognize that in this picture the tRNA has been flattened down in a very unrealistic way, in a two-dimensional way onto the page, to allow us to see that there's an anticodon arm, there's a variable arm that's called that because it can vary in length, There's a T-cy-C arm that has really unusual nucleotides, such as ribothymidine and pseudouridine. They're very unusual nucleotides. Now, the D arm is another arm, and finally, up at the top, we get the amino acid bound at what's called the acceptor stem. And that's where we see the corresponding amino acid to whatever codon is found in the mRNA. So notice that that codon is AAG. And use your amino acid table to look up that codon so that we know what amino acid is bound on the acceptor stem. And hopefully when you look that up in your table, you're going to find that that is lysine. So we'll recognize, looking that up, AAG, and there it is, lysine, that basic amino acid that is carried on that acceptor stem. So attached on to the most 3' end of this tRNA, we get the lysine amino acid. And that's an important note, that this is the 3' end of the tRNA. In fact, that's essential because we know that this must be, the anticodon must be anti-parallel to the codon. So this has to be the 3' end and this the 5' end in order for this to run anti-parallel to the codon. So recognizing that that polarity exists within the tRNA. So the amino acid gets activated when it is attached onto that tRNA. And it's cool the nomenclature that goes along with this. I thought Keenan would particularly appreciate this. It's kind of got this medieval sense to it because a tRNA is named based upon the amino acid that it's supposed to carry. So we'll say then that a tRNA is meant to carry lysine. It's destined, if you will, to carry lysine. And when it's called lysyl tRNA lyse, it means that it is carrying lysine. So tRNA lyse is a tRNA meant to carry lysine, and then lysyl tRNA lyse is a tRNA meant to carry lysine that does carry lysine. Now that we feel super stoked about our knowledge of the tRNA structure and function, it leaves us with the one task of practicing to see if we make good tRNAs. So let's go ahead and begin with a simple example, and then we'll make it a little harder and see how we do. So what amino acid does a tRNA carry if it has the anticodon sequence of 3' GCG? I find that it helps to go ahead and draw out a bit of the anticodon arm to just sort of give the view of what will be happening on the anticodon arm and then the corresponding codon that it pairs with. So there's our anticodon GCG, and it is written in the 3' to 5' direction. So we're in good shape there. We know that the anticodon will travel 3' to 5'. So then there's the rest of the anticodon arm. And then we know the tRNA will pair with the mRNA. So we'll go ahead and draw down the mRNA and see if we can determine the sequence of the codon. So G will pair with C, C with G, and then G with C. So now we know the codon sequence is CGC. So that's what we need to actually look up in our codon usage table and see what amino acid would be carried then up here on the acceptor stem of my extraordinarily ugly tRNA. So let's get out our codon usage table and see what that would be. So look up in your codon usage table CGC, and I'll look it up too. So CGC, that should come out to be arginine. So the amino acid that is carried on the acceptor stem of this particular tRNA is yet the other basic pirate amino acid, arginine. That one was pretty easy, so let's see if we can challenge ourselves just a little bit more. With the idea that sometimes the anticodon sequence is not given in this opposite directionality of 3' to 5'. In fact, sometimes it might just simply be written out without giving the termini. That's a very common thing to see. So if termini are not given, then we always assume that the directionality is 5' to 3'. So let's look at an example of that. So let's look at an example where we would have an anticodon sequence actually that was written in that 5' to 3' orientation. That is to say that maybe the termini weren't even given at all. So let's say that that is UCG. And so nothing is specified. That means we have to assume that the sequence is given 5' to 3', as is traditional to write. So we would have to then remember that on the anticodon arm, so we'll draw our anticodon arm, on that it would have to travel 3' to 5'. So this would be G, C, and then U, and then the rest of our tRNA. So then that would pair with the codon in the mRNA, and that would be G pairing with C, C pairing with G, and U pairing with A. And that's a 3, a very ugly 3. So now we can look up our codon in the codon usage table, C, G, A, and see what amino acid this particular tRNA carries. So C, G, and A. Oh, check it out. It's arginine again. Even though we had a very different, we see the degeneracy in the code with this example. So again, on the acceptor stem of this particular tRNA, we would again see the amino acid arginine attached to that 3' terminus. Awesome. I hope you feel like that's a lot of practice to help you through many of the examples that are on the homework, as well as some that will be found on the exam as well. Let me know if you have any questions. I did notice that one of the things that can be kind of confusing is looking at this structure of a tRNA in the form that it's given when it's flattened straight out onto a page. It's sort of deceiving because, in fact, what will happen to a tRNA is once all those hydrogen bonds form within the anticodon arm and within the T side C arm, the D arm, that means that the tRNA will legitimately take on a three-dimensional structure of its own. And it actually ends up looking sort of like an L-shaped molecule. And this is a picture here that gives a much more realistic picture of what the tRNA would look like. The anticodon arm still down here that would pair with the mRNA and with the codon on the mRNA. And then the acceptor stem, however, would be up here. So this is where we would see the attachment of the amino acid in actuality in a more realistic view of the structure. Now, one thing that's really cool to think about is that there's a tRNA that recognizes the codon in the mRNA. But what's really crazy is remember how there are 64, and actually we know that not all of those, some of those are stop codons, right? But there are 61 different codons that can be recognized within the mRNA. But what's crazy is that there are not 61 different tRNAs. In fact, there's like around 40-something different tRNAs. So that being the case, what's going on? How is it that 40-some tRNAs can actually recognize 60-some of the codons on mRNA? Well, it turns out that tRNAs are a part of the root of degeneracy because there's some loosey-goosiness about what a tRNA can recognize on the codon. There are 64 codons, but there are not 64 tRNAs. Well, we know we have stop codons, right? But still then, there are 61 codons, and there are only 40-some tRNAs. And what that means is that tRNAs must be able to recognize more than one codon. That is, the root of degeneracy comes partially from the way in which tRNAs pair very loosey-goosey with the codon. So looking at a really good example of this is to look at the anticodon CCG. And if we recognize that anticodon and write that onto our acceptor stem, CCG, then what's really crazy about this is that we know that this would typically, in a codon, it would typically pair with G, G, C. And that is one of the codons that it recognizes. But look at this. It also recognizes GGU. So this G right there pairs flexibly with the most 3' position of the codon. Whoa, and that's where degeneracy takes place, right? That is the position where we see degeneracy is in that most 3' side of the codon. So when we think about that, it makes a lot of sense that this loosey-goosey pairing, where G could either pair with C or it could pair with U, is a big part of the root of degeneracy. And so we call this loosey-goosey pairing, we call it wobble. So it's kind of an awesome name for that phenomenon, wobble. And in fact, you know, it just sounds sexy to say that wobble leads to degeneracy. So the next time you're out on a date and you just have no more words to share, just throw out wobble leads to degeneracy, and your date will want to marry you probably. All right, so I'm actually going to pause right there, and we're gonna call it a day for today. And I hope that every one of you has a phenomenal weekend. And don't do anything I wouldn't do. And I'll be in touch, and I'll see you. I can't wait to see you all on Monday.